Thank you for joining this live broadcasting, this live Q&A with Dr. Chernoff. This is a big day for us since we are doing a fundraising to jumpstart a specific research that Dr. Chernoff is going to talk about. To give you some context, we started NF2 Biosolution more than 3 years ago, and we are working, we founded, jumpstarted different NF2 gene therapy approaches. So far, we started gene addition therapy, gene replacement therapy, suicide gene therapy, but now we're talking about a fourth approach that's called gene editing. And we are going to, Dr. Chernoff is going to explain the specificity of this approach. As you know, we don't know yet what's going to exactly work on the NF2 patients. So we have to start different approach and hope that one of these or several of them are going really to work. We have also different goals in mind. We have the goal of destroying existing tumor, and we have also the goal of not having new tumor that are going to appear. So different goals, we need different approach to make sure that at least one or several of them are going really to work for the long term. We need a long-term therapy for all the NF2 patients. My name is Gilles Atlan. I'm the Vice President of NF2 Biosolution. I'm the dad of Karen, that she's 14 years old and she lives with NF2. She went through several surgeries already, and she might go through more surgery next year. And I'm really thanking you for all your donations. We have collected almost $25,000 in the first hours of Giving Tuesday on Facebook. Thanks to this donation, we will be able to jumpstart the project at the lab of Dr. Chernoff. So let me welcome Dr. Chernoff. Oh, we have Nicole also.
Yeah, here. Is it just that I can't see that anybody else is here, or is there actually nobody here?
Nicole, it is broadcasting, it is live broadcasting on Facebook.
Oh, okay.
Yes.
I'm going to take off my video then.
No, you can say hello to everybody, please.
In my OR garb. Hi. I'm going to have to go back in a second. I can't stay for too long, but I'm going to listen as much as I can.
Okay, great. Thank you, Nicole. So please, Dr. Chernoff, if you could introduce yourself and also share your experience in the context of NF2 and solid tumor.
Thanks, Jules, and hi, Nicole. And thanks for giving me the opportunity to join with you today. This area has been a passion of mine for some time. Just to introduce myself to the listeners and audience, I'm a cancer scientist and also a physician. I've been in this field of cancer research for a good 30 years now. I'm the scientific director at Fox Chase Cancer Center in Philadelphia. As of tomorrow, I'm going to be the director of the Fox Chase Cancer Center in Philadelphia. I will still maintain my laboratory. Now, my interest in NF2 dates back about 20 years now. I had and am interested in an area of cancer research called signal transduction, which is how proteins signal to one another within a cell in the context of cancer. And one of the proteins I discovered was an enzyme called PAC1. And at that point, we were interested in this pathway. We didn't really know what diseases it was going to be relevant to. But then a colleague of mine, Joe Testa at this institution, and myself in 2001, just exactly 20 years ago now, found that this kinase interacted with the merlin protein. And I'm sure your listeners today know that the merlin protein is the protein encoded by the NF2 gene, the one that's damaged or missing in the NF2 syndrome. And so once that connection was made between this enzyme I had been studying and the merlin protein, I had to start learning about a disease I frankly knew very little about. And I think that's true for a lot of physician scientists. NF2 is a very important disease, but not a very common one. And it's very difficult, as I guess if I had known in the beginning how tough it was going to be to come up with therapies, I don't know whether that would have turned me away or not. Actually, to me, it's maybe even more determined, and this is true of my colleagues as well, that we got to do something here. And so for the next, since 2001, I've been working at trying to find targets, as we say, in NF2 that would be useful in treating or preventing this disease. Now, after a lot of years of work in this area, both from my lab and the field in general, I've come to the conclusion that small molecule drugs, in other words, in the kind of drugs you normally take in a pill or an injection, on their own are unlikely to be curative in NF2. I can't say that for absolutely certain, but based on the experience that I've seen myself and among others, I think a fresh approach is needed. And so I was very encouraged by this group's emphasis on a totally new way of doing things, which would be some form of genetic therapy. In other words, try to repair, replace the damaged gene, which is, after all, the ultimate cause of NF2, or use something about the genetics of the disease to eliminate the existing tumors, as Jill's mentioned in the beginning. So, fortunately, the tools for doing that kind of approach, in other words, genetic therapy or genetic medicine, have been evolving at a very rapid clip in the last 10 years, and especially in the last 5 years. We now have the ability, at least in a test tube and even in mice, to alter genes and fix genes, if you will. And so I started getting into this area of gene editing. I'll just refer to it that way because that's the main approach I'm interested in. A few years ago, we've had some success in non-NF2 settings in editing genes at will. And we are now about to apply this in the NF2 setting using patient-derived materials to see, can we repair the damage? Can we do it efficiently? Can we do it without damaging other genes? I mean, there's a lot of things that has to go right here. Right? And so that's what we're doing with the support of this group, and I'm happy to discuss any aspect of it with you.
Thank you. Thank you. So let's start with that specific project, CRISPR gene editing. So first, what is CRISPR and how does it work and from where it's coming from? CRISPR gene editing.
Yeah, so the CRISPR gene editing, which is well known enough now to have made it into newspapers and magazines and so on, was the subject of a Nobel Prize 2 years ago to Jennifer Doudna and her colleague from France. Had many sort of discoverers, if you will, like most things in science. I mean, it really dates back a decade and a half or so to work from people in bacteriologists, people studying bacteria, who would notice these sort of strange repeated sequences in the bacterial genome. They didn't really know what they were all about, but they noted that they existed. And later it was shown that they had something to do with resistance of bacteria to viruses. So just like we get viruses, bacteria can get viruses too. And their way of fighting off infection is somewhat different than ours. And one of the ways that they developed was a system that we now call CRISPR, where they can recognize a foreign invader, namely a virus, and activate enzymes within the bacteria that chew up the invader's DNA. So that took a while to work out. It was a very unusual thing that no one had really suspected existed. But the reason that leaped from a kind of bacterial study, which interested people like me, but the general public probably didn't care so much about it, was that Jennifer Doudna and a few others figured out how to adapt this system from bacteria to work in human cells and other mammalian cells, where we could use the system to destroy other strands of DNA, not necessarily viral DNA coming in, which was the original purpose of CRISPR as nature devised it, but to destroy any DNA sequence that we wanted to. And so the original incarnation of this, if you will, was to show that you could damage genes, not fix them. That came a little later, but you take, let's say there was an oncogene that was active in a cancer. This would not be true in NF2, but in other cancers. And you say, oh, okay, I'd like to get rid of that oncogene because it's causing problems in the cell. Well, now you had a genetic means to do it. If theoretically, at least, you could introduce a CRISPR construct into cells. It would find that target sequence in the oncogene, cut it, and destroy it. And that was that's still a common use of CRISPR in the laboratory, and even in some clinical application. But for something like NF2, or any situation where you really don't want to get rid of something, you actually want to restore function, because in NF2, the gene is damaged or missing, not unlike an oncogene, which is overactivated. This is underactivated, if you will. You needed something a little different, let's say, and that would be altering single bases or inserting small numbers of bases into the damaged gene rather than trying to cut it and destroy it. And a variety of people figured out that you could adapt the CRISPR system to do just that. And there's been a number of different strategies on how to do it that's still being worked on even today to improve the system. Basically, every month I read another paper that shows somebody else has made, an advance. But the general idea is you can take any sequence anywhere and alter it to anything. That's not quite true today, but that will be true very soon. And we can already do many of those things using the variety of CRISPR tools.
And is CRISPR today currently used in humans for other disease or very close to be used?
It is in some phase 1 trials now for limited numbers of types of diseases. I mean, one of the ones that's been kind of popular in the gene therapy field has been eye diseases, because there you can essentially introduce whatever your CRISPR reagent is into the eye rather than to the general circulation of the body. And so it's kind of easy to monitor what's going on and easy to control any bad side effects that you might see. So there are plenty of, excuse me?
And easy to access the place where you
Yeah, I mean, so far, again, we're very early days here, but there have been at least, media reports about this approach restoring function in people who have various forms of blindness, et cetera. There are also beginning attempts of doing this not just in the eye, but elsewhere in the body, especially diseases that can be fixed by doing something in the liver. So there's plenty of metabolic diseases that involve mutant versions of proteins that are required for metabolizing various nutrients, or things like thalassemias or sickle cell anemia, things where you don't make the right form of hemoglobin that causes a lot of problems. There are already gene therapy approaches, at least for sickle cell, that have been shown to have some success where they're actually repairing or changing the types of hemoglobins that get expressed in patients and that relieving their symptoms. So hemophilia would be another one, but all of these involve either altering genes that are in the bloodstream or in the liver, because those are kind of easy, relatively easy to deliver the CRISPR to. I think when we talk about NF2 and other situations where you're not in the liver and not in the blood, other issues begin to appear, other difficulties. That is, how do you deliver your gene altering machinery to the right place and get it to work there? And that is a serious issue. I don't want to minimize it. I do think it's an addressable one, but that's one step up the ladder from what's being done now.
And why do you think CRISPR will help with the NF2 disease? Why specifically NF2?
Well, I think it applies beyond NF2, but I think an NF2 is a particularly good candidate for this approach because the protein at issue here, the merlin protein, is not an enzyme. It has very important functions, obviously, or else people wouldn't have problems when it's mutated, but it's not an enzyme that, and enzymes are much easier to deal with small molecules. You can disable them if you will, and sometimes you can activate them, which would be what we want to do in NF2. But the protein that it actually is very difficult to target that way. And so the beauty of CRISPR or any genetic type, let's say gene replacement approach, which is something else than CRISPR that we can talk about if you want, is that you're dealing with the problem directly. You're not looking at proteins that interact with merlin, proteins that are downstream of merlin or upstream of merlin. You're dealing with the problematic protein itself. We can't do it with small molecules, apparently, but we could potentially do it by fixing the gene that encodes it. So I think that's the difference here, is that if for once, after all these years, we can go after the central cause of the disease.
Just some few things to say to all our people that are watching. You can ask your questions by writing your questions below that chat, below that window where you see the live broadcasting. So please write any questions you want. And once we are done over these questions that I'm asking, we will have some time to read your questions as well. So don't hesitate to ask your questions. To come back to gene therapy for NF2 specifically, since we have different type of mutation in the NF2 genes, So what do you think, what kind of, I mean, I know it's hard to answer, but what kind of percentage of NF2 patients can really benefit from CRISPR? Will there be some type of mutation that CRISPR cannot correct? Will there be some type of mutation that will be very easy for CRISPR to correct? If you could expand on that.
Those are great questions. Difficult, but great. So this is not a panacea for every single patient. I don't think this approach will necessarily work in every patient with NF2, but there are definitely subsets where it's a more favorable approach than others. So where it's best suited is when the damage is small, and that means point mutations, which there are plenty of people who have splice site mutations, for example. It might be a single base pair. That's the only error they've got, but it's devastating because if it gets a splice junction, then the gene isn't transcribed properly, the protein effectively isn't made. So just because you have a tiny bit of damage doesn't mean that you don't have serious consequence. Those situations are quite amenable, at least in theory, to the CRISPR approach, because all you're trying to fix is a single letter, a G, we turn it into an A, or an A to a T, or whatever the particular issue might be. But not everyone has point mutations. There are patients who have all kinds of other damage. So the next most favorable would be small deletions. So there are lots of patients, I actually don't have the figure in my head about the percentage of the total, but not rarely, let's say, people will have a small deletion. I'm talking about, 4 or 5 bases. It's usually a number that's not divisible by 3, because if you lose 3 bases and you just lose 1 amino acid, generally that doesn't screw up the protein too badly. But numbers like 4 and 5 or 7 and 8 are bad, because that means the protein's going to be out of frame. So all that sort of technical mumbo jumbo is just to say that people who have these small deletions, which may be just a few bases, are also quite amenable in theory to CRISPR fix. Now, there are people who have much larger deletions. There, it's a little less clear. CRISPR can be used to insert large pieces of DNA. And so we are actually going to be trying that in one of our patient situations. We have a large deletion that we are going to attempt to fix. And there's been plenty of CRISPR literature saying that that can be done. But where it would get, and then the fourth category with most difficult, I would say, and probably not the ideal approach, is for people who have complete loss of the gene. So remember, in people who have these tumors, generally one allele may be inherited, but then the other gets lost somatically. In other words, as the child is growing up, and weren't born that way. That's often accompanied by complete loss of one of the chromosomes or the entire NF2 gene, and then either total loss or one of these point mutations or small mutations in the other gene, the other allele. If both alleles are completely gone, which does happen, then CRISPR has nothing to fix, because there's no particular mutation there to alter. The whole gene is gone. So there, gene replacement is a much more logical approach. Now, maybe CRISPR may assist that in some way, but in general, we're talking about something different there, which is also a very promising avenue, but would be less good for fixing point mutations. So I think there's situations where one is favored and there's situations where the other is favored. And I think we're obligated as both families affected with this, as donors, as scientists, to consider the whole here. And we're not going to craft a single solution that will be right for every single patient. It's going to require somebody to think hard about it and pick the right choice. There may be some where either one could apply. But again, I wouldn't say that CRISPR is going to be the fix for everything.
Yes, to remind our listener, and I said that at the beginning, we are sponsoring and funding different approach. And the other approach that we are funding already for almost 2 years is gene addition and gene replacement. So we are replacing totally the gene. So even if CRISPR cannot take care of specific type of mutation, we have that other approach. And again, that's why we have to go in parallel and do some discovery and research for finding, having a different approach that can, at the end, tackle all the population of NF2 and not only some of them. To come back to CRISPR, what are the risks that we know today? Didn't a doctor went to jail in China about using CRISPR? I'm sure people heard about it or read other things about CRISPR. And that was, I know, a few years ago. But in terms of risk, how it is used in the medical world?
Yeah, there's, whenever there's a new technology like this, it comes with some dangers. Some of them are scientific dangers, let's say, because the technology is not maybe ready for prime time or for human use. Some of it comes from people's ambition. So in the case of the Chinese physician, I mean, this fellow wanted to do germline alterations, in embryos. That may be done someday, but that's like the last thing you do, not the first thing you do, because your chance of damage is maximum. Your chance of helping is, really unknown. And so I think that guy just wanted to become famous and win the Nobel Prize. I mean, I don't know him personally, but it was a ridiculous thing for someone to do and totally unethical. That's why he went to jail. I mean, even in China, which may be somewhat more liberal in how they allow research to proceed, that was way over the line. And so we're not talking about germline alterations here. I mean, maybe someday we will, maybe even in 10 years, I don't know, but we're not nearly ready for that. What we're talking about is somatic changes, taking a patient, say with NF2 or someone with an enzyme deficiency who's already born, we know their problems, they're not getting better on whatever standard of care is. And there we're trying to fix specifically, not whether they pass it on to the next generation. That involves this germline stuff, which I think we are just absolutely a red light for that at the moment. But then there's technical risks. So forgetting about the ethical issues in germline, because we're not doing that, but even applying it to any, you have a teenager with NF2, that person is otherwise healthy, but except for this one horrible problem. Right now, CRISPR is not accurate enough that we can't guarantee unintended damage. So when we do CRISPR, with all of our controls and using the best technology, we intend to fix whatever it is we're trying to fix, the splice site mutation or the small deletion that occurred. We don't want to create additional problems elsewhere. Remember, the genome is large, there's 3 billion bases there. There's a lot of what we'll call off-target potential. And at least in the early forms of CRISPR, there were quite a few of these off-target events occurring, which were fine if you were studying a cell line or even a mouse. Not so good if you're dealing with a human being. So, people have been working very hard on reducing that off-target risk. And there have been great strides. I mean, Jills, we've been in conversation about this, and Nicole also for the last few months, because I know we're all avid readers of the literature. And in some cases, I know some of the principal investigators on these, so I can call them, and, you call them too, I think. But, I'm pretty up on what these folks are doing. And one of the things that all these groups are doing is trying to reduce the off-target unintended effects of CRISPR. And there have been a few approaches that have definitely helped. And one of them that we've talked about in the past is called prime editing, which uses CRISPR technology, but has a very clever twist that involves not completely cutting the DNA. So instead of CRISPR generally makes a double-strand break in DNA, then it fixes it. But if it makes that double-stranded break in an unintended place, it may or may not get fixed there. And there, you create a new mutation. Prime editing minimizes that by not ever doing a double-strand break. It does single-strand breaks, which are much simpler to repair. Even if you do it in an off-target place, it's probably going to be repaired without any permanent damage. That said, even prime editing has a certain off-target rate. But prime editing, we have version 1, there's been version 2, version 3. It keeps getting better and better. So I think the accuracy problem will go away quite soon, where it'll be an acceptable off-target. It's never going to be 0, but it may be so low that we can live with it, because the benefit of fixing the gene will outweigh the tiny risk of causing additional damage. And we're not quite there today, but we're definitely making strides almost monthly. And by we, I don't mean just my lab. In my lab, we're one player in thousands of people out there, but many, many labs who are super interested in this as a general method. So if they fix CRISPR in general and make it more accurate, it's all the better for NF2. Now, the second problem is not so much a danger, but an issue, a really important one, is the efficiency of the process. It's not like, even in a cell line, if I apply CRISPR reagents with my best technology, I'm not going to fix 100% of the cells. I'm just not. And that's in a very controlled environment where I have cells growing in a dish, no body in the way. It's like the ideal test case for a fix. And I mean the whole field, nobody can get 100% fix. In fact, if they get 50%, that's an unusually good result. usually more like 10% or even lower than 10%. That's actually not so much a safety issue, but a more important therapeutic issue, because how are we going to apply this if we're only fixing a small percentage of the lesions? I mean, imagine NF2, you have hundreds of thousands of mutant cells, maybe millions of mutant cells. And I say, okay, I've got this great approach and I can fix 20%. That's my max. That means 80% of those cells never got touched. They're still going to behave just like they did before. Now, the 20% that got fixed, maybe they stop growing so aggressively, maybe they regress, something we can talk about if you want. But we need better efficiency. And that is probably a more difficult problem than the accuracy problem. There have also been great strides there too. It's some of the same reagents used for gene replacement that you referred to, Jill, is about, well, actually we didn't name them, but viral vectors. People use attenuated viruses, in other words, non-disease causing viruses, as a way of introducing this CRISPR machinery into cells, or genes into cells, like the whole NF2 gene for that matter. Those can be used, and it's one of the best approaches we have today, but their efficiency of affection is not 100% either. They're getting better, just like the accuracy issue, but they're not quite there yet so that we could do a trial, say, tomorrow. At best, we could do a trial in mice, what we call a preclinical trial right now, and people are doing that sort of thing. In fact, I'd like to do that sort of thing. But even before that, we have to show that we can get reasonable efficiency in cells, which is the current project.
But we don't need to fix 100% of the cells, correct? But we don't know how much percent we
yeah, that's a very interesting issue. And I know we've debated this, and I've debated with other scientists as well. Like, what would be good enough? So, a cure would require 100% fix. Is a cure, a complete cure, actually necessary at this point in history? Or would it be valuable if you could slow down the whole history of NF2 by a factor of 2 or 4? So, man, as you well know, having children affected with this, it generally takes a long time for the first diagnosis, and then to symptoms, and then symptoms progressing. It can be decades. I mean, it's very unfortunate, but it's slow. If you could alter that timeline so that instead of 20 years, it was 40 years, I mean, that would definitely be of some benefit. It's not as good as a cure, but it's something. And I think the higher the efficiency of the fix, the longer you're going to stretch out things, and the slower things, which means you'd have milder symptoms for a longer time. Again, I realize that is not the ideal or end state, but it would be a huge improvement over where we're at now. I mean, currently we have some small molecule therapies, as you know, a target mTOR pathway or Avastin, things like that, which have, there's some effects, but I don't think anyone would call them home runs. If there was a drug out there today that doubled the span of, say, the NF2 symptoms, people would be, that would be used by everybody for one thing, and it'd be pretty happy that an advance had occurred. I think even with today's efficiencies, if we could, get by the safety issues, et cetera, we could have a significant effect on slowing down this disease, if not stopping it. And I mean, I'd be interested what people think about that, because your families who were affected by this, how much would that change your thinking? Would that be something that interests you or not?
Obviously, for some patients, they need to take care of existing tumors that are already too big, and they need to reduce their size so they don't go through a risky surgery. And some others, and I'm guessing maybe most, will be almost happy for if everything could be frozen, that there will be no more growth.
Yeah.
And so that will be the angle. But again, they think these two different.
Yeah, and that actually brings up something you raised earlier. You said that your group was working on 4 separate approaches. And the one about what to do about existing tumors is an interesting problem. And it's not clear which that different approaches won't work. So certainly one thing, and I know you're supporting this for other laboratories, is a suicide gene approach, where you introduce specifically into the NF2 cells.
And the bacteria approach as well.
Yeah. And right, yeah, bacteria might be a way of doing that as well. And other people are more expert in that than I am. That might reduce the tumor burden, which could be quite helpful. I mean, obviously surgery can reduce the burden, but that has its own complications as well, and depends on how much surgery can a person tolerate. I don't know that fixing the gene, either by replacing it or using editing to repair it wouldn't also eliminate tumor cells. It's not clear because that's never been done in humans before. What would happen if I took somebody who had an existing tumor? We'll just take it down to the cellular level. One cell, a malignant cell in that patient, in part of their schwannoma or their lesion. And if I fixed it, would that cell now just revert to being a normal Schwann cell, or would it undergo suicide and kill itself? I mean, I hope the latter is true. I don't actually know because no one has done that yet. That's one of the things that we're going to be looking for when we get to mice is when I fix it, does the problem kind of just revert to normal or does it go away completely? And I really don't know which is going to happen. So that's a little different. I mean, the suicide approach or the bacterial approach is actively seeking out to kill these cells. In ours, it would almost be a nice byproduct. If we fix it, will it still live? I don't know. I'm guessing it would just normalize and become a normal Schwann cell, which would be fine. But if it got eliminated, that might be okay too. Of course, you don't want to eliminate all of them because you might then lose function that the Schwann cells are supposed to do for neurotransduction. But that's very much an open question. I don't think there's anyone in the world who could answer it with confidence.
So we have similar questions that you might have already answered, questions from Kenny on Facebook. And again, you can ask your questions on Facebook. Right under that live streaming, you have comments, so you can write your questions in the comments. So Kenny was asking, would a technique like gene editing help only patients which don't have severe symptoms, for example, children, or will it help older people too? That has already.
I think potentially it could help both. I mean, in most diseases, and this is also true in NF2, the earlier you get at it, the easier because the burden is less. But of course, the risk benefit ratio is also changed because in a child, they have their entire life in front of them. An older person has less of that life in front of them. So this is not even unique to NF2. It's basically for any illness. And so it would be easier to treat children, but also would be a higher bar to go over in terms of safety because God forbid that you cause more problems than you can solve. Now, older person has their own agency and can make decisions for themselves and may conclude that what's going on with their NF2 is just unacceptable and they're willing to risk sort of the unknown. I mean, we'd never give something that's totally unknown, but there's always the first patients who get things. We sometimes learn unexpected results from them, even going through all the safety tests that we do. So that there's some risk in being a pioneer. In an older patient with a higher disease burden, it's the issues of efficiency of delivery and the efficiency of the repair process itself are going to be more acute. There, maybe we can't fix 40 or 50% of the lesions. Maybe it's 20%. So they might get less benefit because you're only stopping it in 20%. And I still think you might buy time that way, maybe even a significant amount of time, but we really don't know. So, unfortunately, it's not equal. It would be better to do it earlier on if it were possible. Now, at some point, we'll study exactly how much of a difference there is, at least in mice, because there we can decide in a mouse model, which there are some pretty good ones for NF2, what would happen if we repaired when the mouse was very young versus what happened if we just let them get the disease. And they get the disease that's pretty similar to the humans, which is good for a model. What would happen if we tried to repair late? So that's a great experiment to do. We or nor anyone else quite ready to do that yet, but that could be done within the next couple of years.
A question from Eric from Sweden. What kind of gene editing are you using in this approach? Prime editing or another approach?
Yeah, I am.
And why?
Somewhat agnostic on this. It's almost like you can know too little about something and you can know too much. I read all the literature and there's a lot of competing technologies. Some of them aren't even involving CRISPR. There's a whole nother method, which I don't want to get into today unless someone really wants to know about it, that people at Yale have developed, which is another gene editing, gene replacement method that is not CRISPR based. It's quite interesting, but it's only being used by very few people right now. That one supposedly has very few off targets, but you can't do everything. So being one laboratory, I have a number of people who work with me, we have to pick and choose. We can't pick up a procedure, then drop it the next day, unless we're completely convinced that the new method that's coming online is way better than the old method. So I've struggled with this a little bit. We had already developed a very I think, good approach using kind of, I'd say, older fashioned CRISPR, but with some twists when prime editing came out. And so we tried prime editing and it works. Looks like it works for other people. In our hands, it works about the same as the method we were using. So we're in a bit of a dilemma. We can't do everything, but we don't want to use inferior technology either, or something that's going to, work once and then be abandoned by people. Our method uses a double-strand break, which I said before is a little bit risky. It uses single-strand DNA as the sort of template to repair the cut that we make, and that's what fixes the lesion of interest, like the mutation in NF2. We have not seen really any significant off-targets in our experiments now, albeit with a limited number of genes that we've tried. In prime editing, it's a tie for us right now. So even though people are super enthusiastic about it, and we're very interested too, we're going to end up doing both. So, it's like we're talking about at the onset of this discussion. There's probably not a one-size-fits-all therapy, gene therapy for NF2. Even within CRISPR, there's probably not a one-size-fits-all. There may be certain genes or certain types of mutations that are more fixable with prime editing or better fixed. There may be others with other methods. So we have not put all our eggs in one basket on this at this point. We have them in 2 baskets. Now I can't put them in 10 baskets because I don't have enough people to try everything. But I think prime editing deserves a try because it does have some very compelling logic behind it. This other method that we developed, in our hands is equally good, so I'm reluctant to just give it up. I'd like to see how it does in the setting of NF2. We've tried it in other settings. And we'll see, this is the kind of field where I could pick up Nature tomorrow, the journal, and read about some totally out of this blue new approach. I mean, CRISPR was like that, 10 years ago, which is about when CRISPR came on the world 9 years ago. Hardly anybody was expecting this. We were doing gene editing with the so-called talons and something called zinc fingers, that some of you may have heard of, which, theoretically could do all the same things that we're talking about today, but were very inefficient and just hard to make. And then suddenly CRISPR appeared, out of nowhere, and everyone dropped what they were doing and switched over. There is this competing technology I was referring to, out of the Yale group that, I don't know, it'll displace CRISPR, but might end up being just as useful. And there might be things that I don't even know about, unimagined things, because so many people are trying now to edit genes by every means possible. And that's great for people who have kids with NF2 or other genetic diseases, because you don't really care whether it's CRISPR or not. It's just something that will work and is safe. And so that's a long answer to a short question. We're trying 2 different methods is the short answer.
Okay, good. And as I said earlier, we are sponsoring gene addition, gene replacement, suicide gene, and now your lab for gene editing. And all these are genes that has to be delivered. And we discovered that in the last 3 years that at first AAV, virus AAV was a way to deliver, but lately there is also nanoparticles that are not a virus that could be used in order to deliver these genes. So for your opinion, for gene editing, what could be the best, that's another question from Facebook, by the way, what could be the best solution for delivering CRISPR? Will it be lipid nanoparticles, other type of nanoparticles, or AAV virus?
Yeah, a great and very difficult question. And I'm not going to pretend to be like the world's expert on delivery, although, I have some expertise because I use it, but I'm just not someone who develops new methods of delivery. So certainly nanoparticles have been used to deliver all kinds of genetic material, RNA, DNA, proteins, etc., into cells, into animals, into plants for decades. Viral vectors have been around for decades too. Originally, people were using adenovirus itself for gene therapy, and some of you who are old enough may remember the 1999 death of this kid, Jesse Gelsinger at Penn, who was undergoing an early form of gene therapy using adenovirus, and it ended up killing him from the immune reaction to the adenovirus. Subsequently, people abandoned adenovirus as a delivery means and switched to something much safer called adeno-associated virus. So it sounds a little bit the same, but it's not the same. It's AAV, as you mentioned. That one's been used a lot. So there's a pretty good safety profile for AAV delivered material. And that's the one that we tend to use the most. Although in cell settings, we also use lipid particles as well. Now, there are some fundamental differences between the two. with adeno-associated viruses, you can at least attempt to target them to cells of interest. So for example, if I'm delivering CRISPR into someone's body and I'm intending to fix the merlin gene or the NF2 gene in Schwann cells, I'm not so interested in introducing this thing into a liver cell or into a bone cell or into a brain cell or neuron, because that's not the relevant tissue. And so with adenoviruses or AAV viruses, you can direct them to certain tissues. Now, it's almost like putting a zip code on them, if you will. But right now, the zip codes aren't that perfect. So you can deliver them favored to one tissue, but it doesn't mean they don't get a little bit into other tissues. But that's another field that's evolving very quickly. So they're finding out new ways to target the adeno-associated virus to the particular cell of interest. And so right now, we can already favor getting them into Schwann cells. We just can't guarantee that's the only cell that takes them up. I don't know that people can't do better with that. Again, not my field in particular, but I think there will be more specific delivery. Now, the nanoparticles are a bit different. In their rawest form, there's really no targeting. They introduce into the body and they would kind of go everywhere. And so your CRISPR construct would go everywhere and act on DNA in whatever cell it happened to go into. Now that said, there's been improvements there too. And nanoparticles can be engineered so that they favor certain cells by putting various proteins or receptors or molecules in their in the capsule. You can almost make it like a virus, which has a shell around it, which is what makes it go to wherever it's designed to go. It's not impossible that we could make a nanoparticle that would also be targeted to Schwann cells. I mean, the idea behind it is find what is unique about a Schwann cell, what's on the surface of it, especially when it's a cancerous Schwann cell, that could attract a particle that could have something bind to it. If you have such a ligand, is what we call it, you could put it into a nanoparticle, you could have it expressed on the surface of an AAV. The idea behind it is kind of similar, but we don't have a perfect one yet that would satisfy me that it's only going to Schwann cells. So that's another area that needs improvement. I mean, this whole field needs investment. And I don't even mean within just the context of NF2. Within genetic therapies in general, these issues come up again and again. And again, unless you're lucky enough to have a disease that involves a liver enzyme deficiency, where we just inject essentially the DNA into the person or the animal's liver, we need a more elegant delivery vehicle.
And we also need to target meningioma and epidemoma. In addition,
that's true, and they may be separate. It may be that an AAV that works in a Schwann cell, by design, is not going to go into a meningeal cell. I'm not sure about that. It might be the same targeting works for both because they're somewhat related cells by their history, but that's very true. That again, there may be tailoring required for the patient. Now, hopefully, it won't be so individualized that every single patient on Earth who's undergoing gene therapy needs a unique solution. That would be difficult because pharma will never take that up. They'd like to have sort of a common platform. And you can see why. I mean, it's not that they're so evil or anything. It's just that it's hard to control when you have thousands of different types of solutions for each patient. So I think what they're looking for is a platform that may be adaptable, in 4 or 5 ways, but not a million ways. So they want something that would work in the majority of people. I think for the gene replacement, that's more reasonable to do. I think for editing, we'll never get away from having to tailor at least one part of the therapy to cure, to fix the particular lesion, which is different in every patient. So there's no way that a construct I'm making that was tailored to, say, your child is going to work in another child unless they happen to have the exact same mutation. I mean, that's what we're doing here. It's very precise. So that one would require more finesse, if you will, but still, if it works well, and there's only one element that have to change, which is just really a sequence of one part of the CRISPR, it's possible the industry could handle that too. But I think it's easier for them when they have a one-size-fits-all.
Yeah. And let's say if it works, if your experiments work in cells and on animal models, how long do you think, let's say with the proper funding, how long it will take to get to clinical trial?
That's a tough question. It involves so many different layers of both regulatory issues, etc. And I don't want to be a Pollyanna and say it's going to be, in 2 years, because I actually don't think that's going to be true. I think in general, CRISPR type editing it's already being used in phase 1 trials for a few diseases, and it will undoubtedly be done for a lot more within the next couple of years. I think with NF2, because it involves loss of the gene and because it affects children, it's going to take a little longer because of the safety issues. I do think, though, within a decade, there will be genetic therapies, probably beginning with gene replacement, which is, I know, one of the things that you're supporting, probably a little later for editing that come online for technical reasons that we've been talking about during this hour. But certainly within the lifetimes of your children, assuming they're not already, 60 years old or something, I think there could be something for them.
Yeah. Regarding the other approach that we are sponsoring, they're already preparing a clinical trial for the bacterial therapy, and we hope for the suicide gene, it will start soon.
Yeah, where there's more history of similar approaches, the easier it is to do a clinical trial. Where you're getting into unknowns that have never been tried, and gene editing would be one of those. I mean, it is beginning to be tried in some very high-risk situations that are kind of easy pickings, if you will, or low-hanging fruit. NF2 is not that, so I think the editing approach might end up being the best approach there is, but it's going to take a little bit of time until they work out in general how to do this in anybody for any disease. So there's just not a lot of experience out there to date. I mean, bacteria have been used as therapy in one form or another for over 100 years. Not exactly in the form you guys are interested in, but in general, people have been introducing bacteria and phage and all sorts of things way back in the 1920s and before. So,
good. And could you say a few words about your lab, and also if technically it's possible to come to visit your lab if NF2 patients can, if we could organize. I mean, I know nowadays COVID days, some restriction, but let's say in a world without this COVID-19.
So, I mean, assuming that Omicron doesn't shut down everything again, which we're in Philadelphia, so the northeast section of Philly. I'd be very happy to personally meet with anybody who'd like to come up here, introduce them to the lab group. I have a lab. It's getting a little smaller because of my other duties to the center, but I have about 7 people who work with me, and I still do some of my own experiments, believe it or not. They like to see the old guy in the lab, showing I haven't forgotten things, but I enjoy doing that. And most of my personal work is on the NF2 project now because it involves a lot of sort of DNA constructions, which I'm really good at, and I can sort of do them and stop without, problems. If you're doing cell culture work or animal work, you can't be so intermittent as my time tends to be. But anyway, I'm happy to personally show you around both the center and in my particular lab and show you the team that's working with me. Not everyone in my lab works on NF2, but a good half of them do. And the rest are on projects that could be related. I mean, I have a whole group that's working on the hippo pathway, which, I guess we're not going to talk about today, but is another very important signaling element in NF2. And there are some therapeutics in the hippo world, which we're trying as well. We even have some collaborations with industry on that, where they're giving us various compounds. So, it's not just the gene editing. That is maybe the most interesting thing we're doing at the moment, but there are other approaches. And even the PAC project, which is how I got into this whole field, there's some hope. I don't know if people may have seen a paper I did with Wade Clapp earlier this year, where we used a PAC inhibitor in an NF2 model mouse, and it actually did some good in preventing hearing loss. It wasn't a home run by any means, but it was tolerated quite well, and it did slow down hearing loss and slow down tumor growth. So we were pretty encouraged, and I think that may light a fire under industry. I just got contacted by Roche, which is the owner of Genentech, from their branch that's in China, believe it or not. I'm going to have a conversation this Thursday with that team, who's now interested in re-engaging with the PAC project, which they previously worked on. I think part of that is because they saw that paper too and thought there might now be a path forward. So gene editing, I think, is super interesting and important and may ultimately be the best thing we can do, or gene replacement. There's still some hope that we can improve small molecule approaches, which are just easier to get off the ground. And I'm hoping that big pharma will get re-interested in this, because they kind of dropped for a while, in my opinion.
Thank you very much, and we will take your invitation to your lab. before COVID, we had a team, a group of patients visiting the lab at UMass, where they do the gene replacement. We also had a few people visiting the lab of Dr. Kisil in Florida. So I think it's very important to connect the patients to the people working in the lab, to relate to the
in fact, I have to say that I mean, I would welcome even an extended visit. If someone wants to spend a week here, they can do that too. You could work along with my postdoc and actually, I don't know, do some of the work if you're able to use Pipette-Man and know something about science, or at least observe absolutely firsthand. If you want to see our mice, they're there. It's quite an experience. I mean, not for everybody, but I think if someone wants to do that, or if you have a child with NF2 and they want to see what's going on firsthand, I would be very happy to host such a person.
Wow, that's a great idea, to really have some kind of stage or some experience.
Right, you can participate in the work yourself, but then, under a proper instruction, it's not, you can do it. It's not, you don't have to have 15 years of postgraduate education to do it.
Yeah. Oh, perfect. So thank you. I think let's see if we have more questions on the internet. Let's check with Rebecca. Rebecca, do we have any other questions, or should we wrap up?
Not at this moment. I think we can wrap up.
Okay. Thank you. So thank you everybody for joining this live, our first Facebook Live. This video is recorded. Let me put the different view. This video is recorded, so it will be on Facebook, but also a bit later on, it will be on YouTube, so you can watch it again. And I would like to thank again Dr. Chernoff, also for his proposal of being able to come and visit the lab. And thank you also for all your donations that you are giving today, and hopefully in the next following days and months as well, so we can fund this project and make it progress. That's one part of the project. After that, we talked a little bit about delivery. That's another part. Testing on animal models, that's another part. So all these different stage needs funding. So we don't want anything to slow down. So we like always to have this project moving on. So at the end, we have different approach. Since again, as I said, we don't know what's going to work at the end. We need different approach because we also have different goals. So thank you again for your support. And if you have any questions as well, you can still write your questions and we might be able to answer your questions, or we can pass these questions to Dr. Chernoff and he will be able to answer to them.
Yeah, I'm very, very happy to answer anyone's question or participate. So thank you for the invitation and the opportunity.
Yeah, and one last thing, if you do a donation, if you could write on the paper, or let me turn, you don't.
Yeah, you're back on.
Yes. Yeah, if you could write on the paper, never forget to live with hope, and that you donated, and then you do a selfie with it and you post it on your wall, and so it can bring more people to donate to our fundraiser. Thank you so much. Thank you, everybody, and I hope to see you again soon. Bye-bye.
Take care.